Hey guys, and welcome to Lab 6, which is called Enzyme Catalysis. Your teacher might also call this the Catalase Lab, Enzyme Catalase, something like that. Basically, we're going to be looking at the enzyme called catalase and see how it breaks things down. So the first thing that I like to talk about is just the reaction that we're going to be observing. So catalase is going to act on a substrate called hydrogen peroxide, and that's represented as H2O2. And that's going to turn H2O2 into two molecules of H2O and oxygen gas, which is O2. So that's the reaction that we're going to witness today. So I'm going to start off and do part one of the lab for you guys that are virtual and can't do this in class. So the very first thing that we're going to do is add 5 milliliters of hydrogen peroxide to each of our test tubes. So I'm going to do that really quickly. And keep in mind that your hydrogen peroxide will break down in the presence of light naturally into water and oxygen. So you want to move kind of quickly with this. I'm putting just roughly 5 milliliters in here for you to see these reactions take place. So we have a couple different samples that we're going to be utilizing for this lab. We have like a potato, we have a carrot, and we have bovine liver, which is going to be liver from a cow. Okay, so those are going to be the 3 samples that we have of our enzyme, that's catalase, and we're going to see the catalysis reaction or the catabolic reaction. Remember that catabolic, cats have claws and they like to tear things apart. So catalase, ASE, tells you that it's an enzyme, and catalase is going to be breaking down this hydrogen peroxide, and it's going to be turning it into oxygen and into water. Okay, so that's roughly 5 mils in each one of these. Okay, so you can see that we don't have any bubbles occurring, okay? The first thing I'm going to do is introduce our piece of potato. So you can see that we have a little bit of potato fluffing off there, but you have some bubble action happening, right? Ignore all the pool noodles behind me. That's for the model that my kids are going to be building of this reaction. Test tube 2 here, we're going to put the specimen of carrot. Again, you can see that you have some bubblage happening here. I would not rate this as a very high reaction, but you do have some bubbles forming, which means that you know that the enzyme is working. I would say that the potato is definitely bubbling a little bit more. You can see the bubbles up at the top, which tells you that catalase is more active in a potato than in a carrot. And hopefully you can kind of see that there. Okay, and then the last sample I'm going to be using is liver. So I'm going to put that into my next tube here. It's a little nasty. We're going to shove it down there with a stir rod. And that is the liver reaction. So you can see that obviously this had the highest reaction. So what does that tell you? If you have the highest bubbles, that means that you are having the biggest reaction here, the most enzymatic activity happening. So catalase is breaking down the hydrogen peroxide that we added to each of these test tubes, and it's turning it into oxygen, which you can see as bubbles, and water, which is the remainder in the bottom. Okay, so you can see that all of these specimens have catalase activity. I would say that in the bovine liver, you had the highest reactivity. So I would rate that at a 5. On my lab, I rate it from a scale of 0 being no activity to 5, most activity. So I'd probably rate this one at a 5. I would probably rate our potato at maybe like a 2 or a 3 and our carrot at like a 1, right? You can see the little bubble columns in each of these tubes, and you can definitely see that the liver had the highest, which means that it has the most catalase activity. Then the potato, which had the second highest catalase activity. And then our poor little carrot here is not really bubbling all that much, but there are bubbles present, which means oxygen is being released, which means that we do have a chemical reaction going on here involving catalase. So that's the first part of this. Okay, so initially you're supposed to answer when you just have a plain old tube, right? This one I didn't add anything to. Okay, I have a plain tube of hydrogen peroxide here. There's no bubbles formed here. So does hydrogen peroxide bubble without anything present? The answer would be no. When you add one of your substances to the tube, do they bubble? Yes. Why? Because you have catalase that's breaking down that hydrogen peroxide into oxygen and water. Okay. The next thing that you were supposed to do for my lab was to rate them, which we just did. Okay, you're also supposed to touch the tubes. Okay, I can tell you that they're slightly warm. So if they're slightly warm, what does that mean? It means that they're exergonic. They're releasing heat energy. And this one just keeps going. Look at this little guy, right? So they're slightly warm because it's releasing heat energy. So like I said, because there's heat present, it is an exothermic reaction, which means that you're giving off heat. And that's present when we have catalysis. So when we have something breaking apart, okay? So now we're going to test whether the catalase can be reused or not. So we're going to have 5 mils of hydrogen peroxide in an empty tube, and I'm going to pour off the liquid here. I'm just going to get a clean test tube and pour off this liquid. And I'm going to catch the liver if it wants to come out. Okay, so I'm just going to grab that liver that I just pulled out of that test tube and drop it. I'm going to put it into test tube 4, which just had hydrogen peroxide in it. And here's what's happening. So you can see bubbles are forming again. So is catalase reusable, the enzyme? Yes, because we know that when you add fresh substrate to our enzyme, you get a reaction again. And this is because catalase is an enzyme, and enzymes are not used up in chemical reactions. That's why you still have bubbles being formed here. Okay, the next thing you're going to see is if the liquid that I just poured off is, quote, reusable. Okay, so theoretically, if this was present, this is the supernatant that I poured off after the reaction had happened, right? This should be water because the catalase should have broken down all the hydrogen peroxide. So I'm going to add a fresh piece of liver to this test tube, which should be water. Now, keep in mind, there are some bubbles still at the top from the last time. Okay, you see a little bit of bubbling happening, but you can see that like the bottom of that is like pretty still, right? I'm going to go ahead and say that that's mostly water in there, which means that our catalase totally worked. Okay, so is the liquid reusable? No, because it should just be water after the reaction takes place. So that's the first part of this lab that shows you that catalase functions. It functions to break down our hydrogen peroxide into water and into oxygen. So I'm going to reset up for part 2 and I'll be right back. Okay, so now we're going to start part 2 of this experiment, which is going to look at the effect of temperature on the catalase activity. So in the first part, we proved that potatoes, carrots, and liver have catalase activity, which means that they contain the enzyme catalase, which catalyzes the reaction of breaking down hydrogen peroxide into oxygen and water. And then we saw that the catalase is reusable because if you took it out after a reaction was completed and you put it into a new tube with new substrate, which was the hydrogen peroxide, the reaction occurred again, which is exactly what you would expect. And then we also discovered that the fluid left over after the reaction and you put new enzyme into it, nothing happened because that was just water. And catalase does not break down water. It creates water from the breakdown of hydrogen peroxide. So now we're going to look at how temperature affects this reaction. We're just going to actually use the liver because as you saw in the first part, the liver had the highest catalase activity. It's the easiest to see, easiest to measure. So I'm going to be rating the reactions by the height of the bubbles. If you wanted to be more quantitative about this, you could measure in centimeters or millimeters the height of the bubble stacks. I'm just going to be rating them basically 0 to 5. Did they have any activity or did they have a whole lot of activity? So the first thing that I needed to do for this was to put my test tubes full of hydrogen peroxide into different environments. I have a room temperature tube. I have a hot water bath tube. I have an ice bath. And then I also have a piece of liver that was boiled for 10 minutes. And we're going to see how that affects the overall process. So we're going to treat the hydrogen peroxide with different environments to see how temperature is going to affect the rate of the catalysis here. So I'm going to go ahead and pull my test tubes out, and I'm going to do them kind of one at a time for you. So this first one was placed into physiological temperature. That's our hot water bath. It's roughly 37 degrees Celsius, which is physiological temperature. That's like 98.6 degrees Fahrenheit, which is, your body temperature. And theoretically, that's where you should see your enzymes working the best. because that's where they're found naturally. Okay, so the wood's a little bit in the way here, but we have a reaction proceeding here. So this is at the physiological temperature. So this is roughly 37 degrees Celsius. So I'm going to leave that one here. The second tube of our hydrogen peroxide was just sitting at room temperature, which is about 20 to 22 degrees Celsius. So I'm going to put a fresh piece of liver into that one and look at how that worked. So you can see this reaction taking place here. I'm going to let that one sit. Our third test tube was in ice water, and it was about 4 degrees Celsius. And here's my liver sample into that one. So for me, so far, it looks like 1 had the best reaction, 2 had the second best reaction, and 3 so far has the slowest reaction. And now test tube 4, the hydrogen peroxide is room temperature. However, the liver was boiled at 100 degrees. I'm just going to rinse these off real quick. At 100 degrees Celsius. So now it looks a little bit, cooked. So we're going to put that into test tube 4. And here's the reaction rate. You can see that for test tube 4. So you're comparing the differences here. I would still say that 1 had the best reaction, which is exactly what we would expect. Because like I said, this was at physiological temperature, which is the temperature of your body roughly. And that's where this liver should be functioning the best. And then the second one was room temperature, which was 20 something degrees Celsius. See, this one's still going. Look at him go. And then number 3 was in the cold environment, which still has a pretty good reaction, but we'll talk about that in just a second. And then our last one, you see just a couple of bubbles forming, but really I would rate this at a reaction rate of 0. So I would probably rate these at like a 5, a 4, maybe a 3 or a 2, and then a 0. Okay, so that's how temperature is going to affect this enzyme. So like I said, physiological temperature, that's exactly where you would expect the enzyme to have the highest rate of activity, and that's exactly what we are seeing here. And then in the second tube, where you have slightly cooler temperatures, you still see a very good results here, which means that it's active at that temperature, which makes sense. It's not horribly far off from our physiological temperature. 3, you see a good reaction, but you wouldn't expect that at 4 degrees Celsius. So let's talk about a few things here. Remember that this reaction is an exothermic reaction, right? It means it's releasing heat. So as soon as I put the liver, the catalase source, into this test tube, and as soon as it started reacting with that hydrogen peroxide, it's starting to create heat. Also, because I took this out of the ice bath so you could see the reaction, right? Oh, now this one's starting to take off too. Because I took this out of the ice water, now this is starting to heat up because of the temperature around us, right? Now, water does have a high specific heat, and because hydrogen peroxide contains water, it does have a high specific heat as well, which means it resists changes in temperature. But because these reactions are proceeding, they're creating heat, which is going to raise the overall temperature, which pushes it closer to a physiological temperature. Now, obviously, this is it's still a cold environment, which is why it's not reacting as well. But you just saw a few seconds ago that number 2, which was the room temperature, really started taking off. That's probably because the heat of this reaction and the ones from test tube 1 and 2, plus the environment around us, it's kind of warm in here with all the boiling water going on all our hot plates. It's heating up this water, which is causing now this reaction to proceed better at a faster rate. Okay, and then with 4, we have a couple of bubbles, which you wouldn't really expect, but a possible source of this could be, you're using a piece of boiled liver, right? It's definitely cooked on the outside, but did I cut it open? No. So could it potentially still be a little bit raw on the inside? Yes. And this is a liquid, which means it's going to go in and fill in any gaps that are present. So if there's any gaps in this like little chunky piece of liver, the liquid can get into there and then cause a slight reaction. So you see a couple of bubbles forming, but I would still rate this at a reaction rate of 0 because there's hardly any activity, and that's exactly what we would expect. Remember that when an enzyme is past its optimal temperature, it's considered denatured, and especially this, I boiled this for 10 minutes. It's definitely denatured, which means that you've changed the shape of the enzyme. And remember that enzymes and substrates work like a lock and key together. So the shape has physically changed of our boiled liver, which means that now the substrate, the hydrogen peroxide, is not going to fit inside of that anymore correctly, right? So that's why we don't see a very high rate of reaction here, because I've denatured it, and that doesn't work anymore. Okay, so this is part 2. So you can see how temperature affects the rate of the reactions for catalase. And I'm going to set up and show you part 3. So now in part 3, which is the last part of this investigation, we're going to look at the effect of pH on catalase activity. Because remember, this is all about enzymes. Again, you can tell catalase is an enzyme because it ends in ASE. Okay, so catalase is going to be an enzyme, and they are affected by temperature and by pH. That's the environment around them. They have an optimal pH that they function at, and they have an optimal temperature that they function at. So we're going to see, as you deviate from those optimal requirements, how the enzyme activity is affected. So again, I have 4 test tubes here that have hydrogen peroxide in them. I'm going to leave test tube 1 alone. And to test tube 2, I'm going to add a few drops of concentrated hydrochloric acid. So this is going to be like literally like a drop is going to make this very acidic. And then to test tube 3, I'm going to add 1 drop of concentrated sodium hydroxide, which is going to be a base for us. And to test tube 4, I'm going to just kind of wing it based on our pHs of our other 2 that we've changed here. So I just have a glass stir rod that I've rinsed off that I'm going to insert into test tube 1 to grab a little drop. And I'm going to put that down on pH paper. That way we can see the pH of our hydrogen peroxide with nothing in it. I'm going to rinse off my stir rod, and I'm going to dip into 2 and put a little dot on my pH paper and rinse in between. And now I'm going to go into test tube 3 and put a little dot on my pH paper and rinse off. And then let's see, it looks like I have pretty strong acid, pretty strong base. Let's see if I can get number 4 to do like an in-between. So what if I add a bunch of acid and like 1, oops, that did not mix. Let's see if we can stir that together. And like a drop of base. Let's see what happens there. That one is even more basic than it was before. Let me go back and check 3 again. Okay, so those are about the same. So I'm going to say that with my materials here, it looks like I have, based on my little pH, here's my test strip, so you guys can see the different colors. It looks like my first test tube is around a 3, so pH of 3. My second test tube is going to be a pH of 1. So these are pretty acidic. So I guess that the hydrogen peroxide is pretty acidic itself. And then I have test tube 3, which is going to be about maybe an 8, so just over neutral. And then test tube 4 is going to be more like a 12. Okay, so I have like mild acid, a little bit stronger acid, a little bit more neutral, and then pretty basic. Okay, so I'm going to go ahead and add my liver to each one of these. I'm just going to shove them in there and then I'll push them down. So we can see the rates of reactivity here. I'm going to rinse this off in between. So I just have it wet with a little bit of water, which is neutral. So again, that one is like about a 3. That one was about a 1, or on this it has a 0, which some pH scales have and some don't. That one's very acidic. 3 was a little bit more basic, but close to neutral. And 4 was very basic. So based on what I'm seeing, I see that the reaction rate for 3 so far is the best. So I would say 3 is the best, and then maybe like 1 and 4 are about tied, and 2 is the worst reactivity. So I'd maybe rate this at like a 5, and maybe like 4s or 3s, and then maybe like a 2 here. Okay, so does that make sense? Let's think about it. So in your body, you have certain organs that have higher acidity, higher alkalinity, but a lot of the tissues and organs have a neutral pH. So the 1, 3 here, see it's still going. That one was about a pH of around 8, which is very close to neutral. So that one has the best reactivity, which makes sense. Okay, the others, they were, this was pretty basic. This one was pretty acidic, but not the strongest acid. The strongest acid definitely was not an optimal environment. But we can see that we at least have catalase activity, all enzyme activity is on a curve. So even though these are more extreme on the pH scale versus our 8 here, which is, like I said, is pretty close to the middle at neutral, we still see activity here, but it's not as high as number 3. So again, that one was about a pH of 8, which we can see that that's where it functions the best. And then 2 would be like, towards the tail ends of that, like the bell curve for our enzyme activity. So 2 is definitely the most acidic environment, which we can see is not the optimal environment for this enzyme, which makes sense because you don't have a whole lot of acid in your body where you would find catalase, right? So this is how pH affects the activity of catalase. We looked at how the temperature affects the activity of catalase. And at the very beginning, we looked and saw that all of the sources that we talked about today did have catalase activity, but the liver had the highest catalase activity. So that's what we used for the remainder of the lab. So I hope that this helps, you visualize a little bit. Like, we tell you as teachers, as professors, we tell you enzymes are highly affected by their temperature and by their pH. And some enzymes are a little bit more picky than others, that if you have like 1 or 2, pH levels off, it's not going to function even a little bit. Catalase is it's a pretty friendly enzyme. That's why we use it in a lab like this. So you still get to see results. But there are some that are like, if you're off even by a couple of degrees or 1 or 2 pH levels, then it's not going to function at all. But here we did see functionality, but obviously the optimal functionality was obvious and was exactly what we would expect, having it be from a liver from a mammal, right? Okay, so I hope that makes sense. If you have any questions about this for my kiddos, make sure that you let me know in class. For my virtual kiddos, if you have any questions about this, please email me or send me a Remind 101 as well. All these lovely pool noodles are back here because on Friday, you guys are going to be building 3-dimensional models of this reaction. And then on Monday, you guys are going to be doing your video lab reports. So make sure that you watch this. Make sure that you fill out your lab sheet with your post-exam questions. And y'all have a great day.